Sorry, I didn't know I was going to be on camera this morning. My name is Alicia Wenzel. I work with the Surface Water Ambient Monitoring Program at the Central Valley Water Board. And today we'll be hearing a couple presentations from Mauricio at Source Molecular, one of the CEOs who's been involved with source tracking for many years, as well as Yiping Zhao, VP of Technology at Source Molecular, and formerly working at SWRP. So I don't know who all is on board. I know for the water boards, we're all very interested in source tracking science because of the, I think, hundreds of water bodies currently listed in California for indicator bacteria. So we need more information to know how to move forward with those water bodies to find solutions. So thank you, Mauricio.
Thank you.
Oh, yes.
Okay, so thank everyone for attending. The main goal of this meeting is to go through two main presentations. One is going to be sort of like a macro level assessment of where we are with the use of genetic technology in water quality. That's where I'll start with. And the main driver for this originally, and Yiping will go into this in more detail, is the development of a new objective framework for analyzing this kind of genetic data. It's been a major challenge. It's been sort of like the last challenge of using this technology is how you actually interpret all the data. You can have a great lab that produces wonderful data, but then what do you do with it? How do you convert it into something meaningful to improve water quality? And so we'll start here with sort of a macro level of where we are right now with this technology. A little bit of background on our company. So we're Source Molecular. We're a biotech company that was founded on the idea that genetic technology can really move the field forward with respect to water quality improvement. There was a lot of challenges, and there continue to be challenges with current technologies, and we see a tremendous value with this technology. And so it's really two main areas that we focus on. One is our core laboratory. So we're an accredited laboratory. It's actually the world's only accredited microbial source tracking laboratory in the world. And we have started developing project and site analytics. And that's what I was talking about. You know, how, and this was a major challenge for a lot of our clients, how do you actually take this technique and translate it into something tangible for a site, for a project, not just a single sample. Some of the features that we have in our laboratory are digital PCR. This is a new technology. It's like the latest version of PCR. It's more sensitive and it tends to be more accurate in certain instances. We also have a full BSL-2 laboratory that allows us to do pathogen testing. Ends up being important if you're going to try to do some kind of QMRA or natural source exclusion. And then through a partnership with a laboratory that we spun off of, we're doing nutrient source tracking now, which is an isotopic-based technique that can, like microbial source tracking, help you pinpoint the sources of nutrients, whether it's nitrogen loading or phosphate loading. And the latest thing that we've been working on, which we'll talk about more today, is the host fecal score, an objective analytical tool to interpret genetic data. Down. Okay. So the water board is under, from what we've observed and talking to a lot of people, tremendous amount of pressure from different directions. You have pressure from the dischargers, from ag, that are telling you and often worried that they're being blamed incorrectly, or not oftentimes they're saying that they're blamed incorrectly, right? It's always somebody else. And so in some cases it is. In some cases, a city has a tremendous financial burden to fix infrastructure. whether or not they're actually responsible, because there's a general indicator that says there's a presence of pollution. There's pressure from big NGOs. You know, I saw a recent article, the California Water Board being sued by an NGO as recently as last year. So there's pressure from both ends, right? From the people saying enforce more, from the people saying your enforcement is too aggressive. And then there's this underlying pressure from the general public. You know, as we move forward, we're seeing that water quality and clean water is seen as almost it's a human right. You know, and so the community, the city, the citizens are expecting that the state is going to be able to provide them with this water quality. And so there's sort of like this increasing pressure that you guys as a water board need to get this right. You know, and so when you're in this situation, I thought a lot about this in preparing for this presentation. And you go to the next slide. I was thinking, you know, who else is in this scenario of a pressure of getting things right? So we're looking at other industries. You look at forensic science. In that case, somebody's going to death row. Maybe not here in California, but where our lab is based in Florida, someone will go to death based on the decisions made by those organizations. That's a high stakes situation. So what did they look for? They saw that when you're in that scenario where it matters, where you need to make the right call, they've moved to using genetic technology. It's now the gold standard, according to a recent paper from Cornell, in the entire field. That's the kind of impact, the resolution genetic technology provides. Looking at food testing, right? I have an 8-month-old baby. You don't want that food tainted. You don't want that child to die. It's a high stakes situation. They're using genetic technology. When it is a high stakes scenario, people are really moving towards genetic technology because of the high resolution, because of the effectiveness of what the data provides to them. And so we're seeing that the trend nationally, and we've seen this in other countries that are moving towards using genetic technology like Singapore, Hong Kong, is towards the use of this technology as a high resolution tool. So go ahead, next slide. So specifically for a state regulator, a water quality regulator, what is this useful for? Well, we've identified 3 main sort of categories or subcategories and projects within these, but more broadly, the 3 things that people are really interested in: where's the pollution coming from? In that case, you have a lot of culprits, you have a big geographic area. It's sort of confusing. What is actually happening here? Just give me some idea of what's happening. FIB is everywhere. I don't know what's causing it. I need some starting point. That's really been the sort of beginning, and most of the volume of MST is that answering that question. But there's also who's responsible. You know, as a regulator, you have to, in some cases, designate some kind of responsibility so that we can address these problems. And so is it ag upstream? Is it the city? Is it wildlife? Where are we going to designate responsibility? And so that's been a second, more prominent utilization of this technology. But the third, probably the newest but most promising for regulatory agencies, how do we effectively evaluate the BMP effectiveness? So in this case, you have somebody, they said they're going to fix the problem. Let's say farmer saying, it's not us, you do MST, you say it's from them. Then they go back and they say, okay, we put up fencing, we're done. The FIB is still high. How are you going to measure specifically the impact from that BMP on that watershed? If FIB is still potentially going to be remaining from environmental sources, natural sources, wildlife. It's a really challenging situation to be in, because either you're going to say the fence is there, that's good enough, you're okay, which is one extreme, or you say it was positive before, you spent $100,000, $500,000 on fencing, it's still positive for FIB, you didn't do enough. Whether or not they did remove their contribution. And so this is very unclear, this is a very difficult situation, and this is a really powerful tool application for this technology. And so thinking about where we are right now and what tools are available, well, one of the most common tools is the use of legacy testing like culture-based monitoring. This is what's being used for regulatory monitoring, and it's good for that. You're testing, you see there's an exceedance, you go through the entire regulatory mechanism. But when you're doing a source investigation, when you're trying to answer those key questions, it ends up being very challenging. That's because this technique is ineffective at determining and discriminating between sources. So if you have a high hit, it could be coming from animals, from human. It can even be coming from plants. We've seen scenarios where grass clippings can have high levels of E. coli, even though there's no pathogens, there's no health risk. And there's also soils and biofilm. And so these can be complicated situations to manage when you only have this available in a toolbox. So one of the other things we've seen very popular is just field observation, right? Get on the ground, boots on the ground, walk the watershed, figure out what's happening. And you may get a picture like this in your report, and you're saying, it's the cows. That's it. But is this enough to shut down a farm? Is this enough to require action? Is this enough? And in often cases it's not, because the farmer can say, that was 1 cow, 2 cows. It doesn't necessarily mean that this is definitively the cause of the systemic fecal problem across the entire river network. And so the problem here is it's subjective, it's circumstantial evidence, and it's very difficult to defend. It could be one line of evidence that you can use to build a bigger case as to what's causing the pollution, but as sort of a standalone solution, it actually is it can be very weak as a single source of evidence. So what is the consequence of the current techniques, the current tools available? Well, one problem is that it hinders source abatement. The first part of identifying sources of pollution is figuring out what the cause is. And so it's very difficult. In some cases, it's not possible to do that with current techniques. And more importantly, it weakens what we call the chain of inference. So to look at the assumptions that are made, the basis of monitoring is that you have fecal bacteria, right? That fecal bacteria is implying that you have waste, any kind of waste. But that waste is assumed to be human waste. That human waste is understood that it's going to have pathogens. That pathogens are going to cause disease. So it's this chain of inference that you need to monitor, to regulate, to enforce. But this completely breaks down if all these assumptions aren't met. If the bacteria is coming from grass clippings, that breaks down. If the bacteria is coming from gulls, that breaks down, because the risk changes from the different sources. And so this really is a strong, this has a strong consequence on how you're monitoring. People know this, and that's why in the bacteria provisions recently, they've allowed for, and in 2012, the basis of this at the EPA allowed for natural sources pollutions, to state that if the pollution is coming from natural sources that are not controllable, there can be regulatory alternatives to a site. Go next. And so where we are right now is a new emerging technology that's been developed over a decade now, and that's microbial source tracking. So the basis of this, I mean, some of you have some varying degrees of knowledge, but in principle, there's special microbes in the guts of animals that are only associated with them because of diet or the conditions in the gut. There's different microbes in cows than there are in humans than there are in birds. And so through a series of academic exercises throughout the country, there have been genes discovered that are associated with those microbes from cattle and from bird and from geese and from a number of different sources. And at least in our laboratory, we have 13 different animal classes that we've identified and started using through a number of partnerships with different academic institutions throughout the country. And so what this does is it provides a set of methodologies that can identify the sources of fecal contamination. And so where we are right now, you know, probably you may have heard of microbial source tracking over the decade in one article or one publication, but sort of to kind of summarize the last decade is that this technology is actually very mature now. We've developed through a lot of academic exercises the framework to actually start applying this in implementation. And so one of the things that's important, precedent, right? No one, when you're actually trying to apply this and you're not trying to publish papers, wants to be the first one out the gate, right? But this has been applied in waterborne CBI projects. It's been applied in projects throughout the country. As an example, in Boston, but in the case of our laboratory, we've been working with projects in over 40 states now throughout the country where they've been using this technology for different applications. And so there's a tremendous amount of precedent in the utilization of this technology at this point. There's not credible tests. Yeah, go ahead.
Can you define CBI? Some of us are three different backgrounds. What's CBI?
Yeah, I don't know if you want to.
Clean Beach Initiative. It was a big initiative part of the state water board.
Yeah. Yeah. So the test, there's actually very established, standardized tests that have been developed. So the California EPA funded a fairly large project. It was like over a million.
Yeah, it's a source identification protocol project. So it's a big CBI grant that was, the primary goal is to evaluate all the source tracking method that's out there and to come to consensus which ones are the best. So that was the SIP study.
Yeah. And so this created sort of a set of norms of which markers the state should be using and validated those markers to say, look, these are credible tests that can now be applied in implementation. Access to technology, there's now laboratories available. In the case of us, there's even accredited laboratories. There's not a state level accreditation at this point. And so for a laboratory to be accredited, you have to move to like national or international standards where you show the proficiency of running these technologies. And the one piece that has been a challenge, and it's been actually a challenge for a number of our clients for a number of years, is a vehicle for objectively doing the analytics of the data. And so we'll be talking a lot more about that in the second part of the presentation through the host fecal score. This is one tool that can help a number of applications. There are other tools that are being developed right now that we can discuss to kind of fill out a bigger suite of applications. And so just to give sort of like a broader watershed management view, I was thinking about this as I was talking to one of my colleagues, and we went through the different projects that we've done throughout the country. And microbial source tracking, what we found is that people have been able to apply the method through the different stages and not just implementation. And so we've had projects here in California that during the monitoring stage of listing sites and the assessment, they were using this technology to build the case that the watershed is not only polluted, but polluted with anthropogenic sources to strengthen the justification for adding a site to a 303D listing. There's also, during the TMDL, which is the bulk of where we're doing work, guidance as to dischargers, helping them with their waste load allocation, showing that they're doing things, showing the effectiveness of their BMPs, and then obviously through implementation, there's a number of places. But what we did is we sort of identified that the technology is not limited to only one stage in the process of running the Clean Water Act, but there's actually a number of stages where there can be benefit. And it's actually a testament to our customers that have seen that this is really a platform that can be applied at different phases of the Water Act. And so just talk about three kind of more concrete examples from throughout the country. So in Florida, this was a case of site prioritization. So the Florida Department of Environmental Protection in Martin County, it's a few counties up from Miami, they went out, they had high fecal bacteria, inland estuary impacted by bacteria TMDL. It was principally septic system throughout the estuary and the properties. So they did an assessment. They said, okay, we should probably switch the entire system to sewer. Let's see how much that would cost us. So they did hired a consultant, did the assessment. They showed that it was going to cost $138 million to switch the entire system from to sewer. and obviously that was not practical for the county, so they set up a 100-year plan. In 100 years, they're going to switch everything over. But the problem here is, where do you begin, right? How do you eat the elephant, right? And you have to take the right bite to have an impact, at least in our lifetime, on the water quality. And so what they did is they said, okay, we need to rank these sites. So how do we rank them? Did we just use FIB? But they knew clearly there was going to be challenges. They could see the birds flying around in the estuary. They could see the pet waste, potential issues, right? And so they were struggling on how they were going to prioritize. And so what they did is they set up a number of factors, but among them, the most prominent was the presence of the human genetic marker to rank the sites. And so it was very interesting because across the 25 different geographic areas, they were able to actually identify 2 principal areas where they were going to have the biggest impact on the human waste in the estuary. And so by using this tool, they prioritized, and now they're in the process of driving the construction to prioritize based on where it was going to have the best impact on human health in the watershed, rather than simply other, sometimes politicized, at least in Florida, politicized justifications for where they were going to do these septic tanks. I mean, sometimes it could be even financially driven, wealthier communities, let's go there first because they're going to be able to pay for it. But if the focus was on water quality and human health, this was a much more effective mechanism to prioritize where they're going to do their BMPs. And so another example I brought up briefly is Boston Water and Sewer. And so they have a consent decree with the US EPA. They have a systemic problem. They have their MS4 IDDE program. This is actually a really breakthrough project. They were the first program to integrate DNA markers into the site. And so what they found is that the outfalls were, rather than selectively tested, they were measured regardless of the completion of the IDDE. And what they discovered is that the conventional tools that were used, these test kits, field test kits, they were not sensitive and they were not specific at detecting illicit discharges. There was a tremendous number of false positives or false negatives in this site. And so what they discovered is that by using this genetic marker, they can improve the effectiveness of the program and identify clearly where there was human impact, rather than problems where they identified pigeon droppings or pet waste in the site. And so they were able to find better reduction processes by prioritizing within the geographic area of Boston where they had the highest impacts of human pollution. And so our consulting friend Geosyntec, we're very proud of this. They won an award, apparently they're in Napa Valley next week, to recognize the work that was being done. It was really a breakthrough project for the use of genetic technology within an IDDE program. And so I think it was a fantastic execution. You know, we're really a platform company providing a tool, but it's up to the end users how they can creatively implement this technology. And so one project that maybe some of you, to varying degrees, are more familiar with, this is here in California. The Santa Barbara beaches did an investigation. They had infrastructure source investigation. But what did you think? Can you have the narrative of this a little bit more?
Right. So this was a Clean Beach Initiative funded project. So basically, the project that they use microsource tracking technology to go in to find out what are the major contributing sources to the fecal pollution at the beach. So once they found out that I think dog was one of the contributors. So what they did is they implemented a non-structured BMP. They just did an outreach program, going door to door and educating people, collecting after their pets. So, and then they did another evaluation using MSD marker after this implementation of the non-structured BMP. They showed a significant impact of this outreach. So that this was the first study that used this technology to concretely demonstrate how effective a particular BMP practice is.
And so thanks to the kind of resolution of showing that the dog marker was present and then reduced or removed from the site, what we've observed is that, just using the fecal indicator bacteria, it would have been unlikely to show the impact of a specific BMP unless you're able to eliminate every single source and reduce the sort of net level of fecal bacteria. And so this is where we are right now with the technology. This is a real macro assessment, but the technology is really shown to be a superior tool for water quality monitoring because of its high resolution and because of the clarity that it provides that is sort of masked within these more general methods, whether it's ground truthing, which is important, or the monitoring tool of doing culture-based FIB. It can be used to more effectively do investigations of bacteria impairment. And what we're moving towards, and Yiping will now talk about a little bit further, is that it builds a data-driven approach on top of the DNA data to actually execute on these implementation stages by assessing on a project level what the genetic data is actually revealing about the site. That's it. As my contact details, and online there may be some questions that they can chat in. We can kind of go through those, or there's questions to hear it internally in the room. We can talk for about 15 minutes or so.